I just want to give you a very brief overview of what we call the proteomics capacity within CellX, particularly at Aarhus University. Just so you have some idea of where we are. So we're situated in the Department of Biomedicine, not in the new school building, but in what we call Biomedicine South, which is where it houses all our proteomics equipment. I'm not going to talk at all about my specific line of research today, except to say that what we actually do is we look at the contribution of specific membrane proteins and the signaling mechanisms that regulate their function in diseases such as hypertension, chronic kidney disease, electrolyte disorders, and so forth. In doing that, we have what we call our research toolbox. We sort of call it our molecule to man, because we actually study these membrane proteins both from a genetic level, DNA and RNA, all the way through to animal models, through to looking in humans using clinical trials or patient biopsies. For the purpose of today, I'm really focusing on really looking at our work at the protein level and, of course, at a cellular level and single cell landscapes. The major tool that we use, without going into details, to look at the proteins in single cells is so-called shotgun or bottoms-up proteomics using liquid chromatography coupled with tandem mass spectrometry or LC-MS-MS. Chi will talk a little bit more in detail about this, but very simplistically, what is bottoms-up proteomics? We take a sample, it could be a single cell, basically break that cell open to release all the contents, all the proteins. We then chop those proteins into very small pieces, so we digest them using enzymes, and then we separate those individual peptides based on their hydrophobicity and other chemical properties using HPLC. And then each of these separated peptides basically gets fragmented or broken into very small backbone peptides where they have the different mass over charge ratios, and this mass over charge ratio can be detected using the mass spectrometer, and then basically this is then matched against a theoretical database of proteins that are already already digested, then fragmented, and from that we're able to identify the individual peptide and therefore the individual protein. So what equipment do we have access to, or what equipment do we use for our proteomics? Well, we have what we call our workhorse system. So this is a nanoflow EZ-LC system coupled to an Orbitrap Fusion tribrid mass spectrometer. Again, I won't go into any of the technical details about this, but it has basically 3 types of mass analyzer: a quadrupole, an ion trap, and an Orbitrap. And what does it basically enable us to do, or what's so good about it, is it gives us this wide dynamic range and very high resolution for the peptides and extremely accurate mass data. It also has relatively good sensitivity depending on the settings that you use on the system. And for us, what's very important is it's highly reliable and easy to maintain. The newer system, which was partly funded from the CellX initiative, which we're tailoring towards single-cell proteomics, which we'll talk about later, and especially Chi will talk about, is an Orbitrap Ascend tribrid mass spectrometer with a different type of HPLC, which is also coupled to what's called a FAMES Pro Duo interface. What this is really is a way of having additional peptide separation method that basically enhances the sensitivity and detection limits and uses gas phase fractionation of the peptides. And Chi will talk about this a little bit more later. Again, what does it do? It gives us extremely high sensitivity down to a single cell level. It has some other sort of specific things that allow us to be quicker and to basically set up programs and run samples much quicker. And it also has a very large dynamic range. Of course, coupled to the equipment, we have access and knowledge and skills and an awful lot of databases and software that we use to analyze the proteomic data. I won't go into the details of them, but anyone that runs through the CellX initiative or collaborates through that will have access to the same software and help with it as well. So just some simple examples of some basic approaches we can use before tailoring it towards spatial proteomics and single-cell proteomics. For example, we do quantitative proteomics, so we use various different approaches for relative and absolute protein quantification. This can be from animal models, it can be from cells. So we can basically label proteins themselves at a cellular level, for example, using metabolic labeling. We do quite a lot of this. But really, a lot more of the work that we do these days focuses on so-called isobaric tagging. So this is basically adding, for want of a better term, barcodes to individual peptides that allows us to identify which sample these peptides came from when we make a mixture of samples before mass spectrometry. And Chi will talk about that more again in detail in a minute. An example, just in a very generic way that you can use these approaches. You could take, for example, a mouse model that had specific fluorescence in an individual cell type, for example, GFP fluorescence. You can see here just an example of an antibody labeling of a kidney section. And in the same cells, we have expression of GFP. And then we're able to take tissue and take it to a single cell level by digestion. And then, for example, using FACS, we're able to individually isolate the fluorescent cells and then put those fluorescent cells into what we call a TMT-based peptide labeling strategy. So here, basically, every single protein sample from an individual animal gets barcoded with one of these TMT isobaric labels. We can mix all those samples together, and then we can run all those samples together on the mass spectrometer. And at the very end of it, we're able to pull out quantitative data, but we're also able to know which animal or which sample these peptides came from in the first place. And this technique has been applied to single cell proteomics in the so-called SCOPE MS, which again will be touched upon later. And of course, at the end of it, we apply various bioinformatics and computational methods to look for various signaling mechanisms that are specific for individual cell types. Another thing that we can do, again, in a more general sense, is we do a lot of post-translational modification identification and quantification of these modifications. Again, we tend to use our workhorse system for this approach. And what we're able to do is take various samples, and we have a variety of different enrichment strategies for modifications, so we can enrich the total protein contents of the sample for phosphorylation, ubiquitination, sumylation, many other different things. And then again, we're able to use the mass spectrometer to specifically identify individual residues on proteins that have, for example, phosphorylation sites or have other modifications. An example at a protein level would be, for example, this protein here, relatively long membrane transport protein, where again, using PTM analysis by mass spectrometry, we're able to identify specific lysine residues that are ubiquitinated and then look at their function. And we can combine these kind of approaches with the TMT labeling approaches or other labeling approaches to get quantitative measurements of the different modifications. So a little bit then tailored towards CellX, a little bit tailored towards single cell proteomics is what we call small scale spatial proteomics. And to do that, we make use of a laser capture microdissection system. Those of you that don't know what this is, basically it's a very high resolution microscope that we're able to both look at tissue, and we're also able to cut pieces of tissue and drop them into a sample holder. And on your right here in the movie, you can see an example. This is a fluorescently labeled section where we're basically looking at the cells of interest. We're drawing around them very quickly. And then what we're able to do once we've identified the cells and what we want to cut, we basically set the system going. And the very high resolution laser is able to cut at a precision of about 0.5 micrometers. It cuts out the tissue falls into the collection vial below. And then from that collection vial, then we're able to go through our proteomic workflow using optimized techniques that don't, for example, make use of detergents or other things that we don't want to go into mass spectrometer. And again, if we go to our workhorse system and then use ultra high sensitivity settings, then from about 100 cells that are isolated from archive formaldehyde fixed paraffin embedded tissue, and this is a key point that we can actually use archive tissue that may have been stored for many years. we can get approximately 1,500 proteins from around 100 cells using this approach. So that's sort of so you can get spatial information as well as the protein information. And then moving specifically towards single cell proteomics, again, this is the area that Chi will talk about it in his talk, but what we've been aiming to do is basically to increase automation and throughput of our proteomics platforms. One of the things that we do for that, or an approach that we use, is we've had to develop novel approaches for basically doing in situ sample preparation. One of the things that we use for that is this what we call non-contact acoustic liquid handling. And basically what that allows us to do, it basically uses sound waves to push fixed volumes of reagent and fixed volumes of sample out of the machine in volumes around 8 to 150 nanoliters and very high precision, around 0.1 nanoliter precision. And it's very, very fast. We can do about 100 droplets per second, which means it takes about 7 seconds to do a whole plate. And we're able to basically put different reagents into the wells and process the whole cell content within one well of a microplate and not move it between wells where we know we lose samples. And the system is able to basically use lasers to make sure that you only put one droplet into each individual well, and so forth and so forth. Using that and coupling it again to the newer mass spec system, we're able to identify around 2,000 proteins that could be identified from a single cell. So around 250 picograms of protein, we can identify this upwards of 2,000 proteins. This system, although I'm not talking about it at all today, has many other capabilities. For example, we can do multi-omics on it. We can do a lot of TMT labeling up to 18-plex. We can do top-down proteomics, which we won't talk about at all. We can do native omics and so forth, but particularly and specifically for CellX, it's tailored towards single-cell proteomics. How much does it cost? Well, that's something that we are still trying to work out. So I can say that it's not cheap. So if you take into context, for example, service contracts for equipment of around 5,000 kroner a week. If you take into the columns that we use for the HPLC, you can see they're upwards of around 20,000 kroner for a single column. The consumables added onto that, labeling kits, the actual runtime on the mass spectrometer, and of course the data analysis at the end, and the access to the software that we have to subscribe to, of course, all adds up. But over the next 6 months, we hope to be able to come up with a ballpark figure for how much it would cost to process, for example, a 396 well plate with single cells in each well. The future, this is where we hope to be heading. We need to improve on our automation and cell sorting. One way to do that is this new Cell-in-One system, which basically has an ability to sort single cells with 100% accuracy, so you don't get doublets or cell debris. The sorting is based on bright field or fluorescence, and it is quite a sensitive approach. So it's actually able to sort fragile cells, such as certain types of stem cells, and it has a very low sample handling capacity, so you can work in picoliter ranges of volumes, and it's relatively high throughput. Also on the market now for the last 3 months is a new mass spectrometer itself, which is a Orbitrap Astral mass spectrometer, which again, without going into the details, is a super sensitive system, and more important maybe than the sensitivity is the fact that the sensitivity is coupled with high throughput. So basically it takes the time down to analyze a single sample from approximately 45 minutes down to approximately 10 to 15 minutes. So basically the throughput of the whole platform is increased. If you're interested in basically putting a pilot project proposal in through the CellX platform, you can do that on the website that you can access freely, or you can contact myself, obviously CellX manager Lei or Chi, who will talk in a minute. And that's really my introduction to the single cell, and Chi will take it from here. As well as CellX, I also want to thank especially Carlsberg Foundation that have actually provided a lot of the funding towards some of the equipment that I've told you today. And with that, I'm sorry for the delayed start, and I will pass over to Chi, who will go more into the actual data we're generating using the single cell proteomics.